In our initial overview of the skeletal system, we mentioned these general functions. Support implies shape, and indeed, this organ system provides shape to the body. Protection, well, critical vital organs are behind bone. For example, your brain is housed within the cranial cavity of your skull to protect this vital tissue from damage. Even the heart and lungs are behind your ribs. Movement. We use skeletal muscles to move some bones against other bones. For example, nodding your head yes or walking. Mineral storage. Most folks are aware that calcium is stored in these organs. Blood cell formation. Red bone marrow is housed within these organs. Cartilage is associated with the skeletal system. As a result, no conversation about bones is complete without discussion of cartilage. Hyaline cartilage possesses a hard matrix, resists compression, and provides shape to some organs. As discussed in lab, we can find this tissue in the respiratory system, nose, ribs, and long bones. Fibrocartilage contains a different fiber in the matrix. This tissue absorbs compression and provides cushion and shock absorption between bones. For example, the menisces of the knee prevent the femur from hitting the tibia when we stand, walk, and jump. Also, the fibrocartilage pads between vertebrae cushion each vertebra but also position the vertebra so that nerves can pass between them without being pinched. Elastic cartilage is found in two places and, as its name implies, exhibits stretch and recoil. The outer structure of your ear, the pinna, and the epiglottis both contain elastic cartilage. Here we see the locations of the three cartilage tissues in the body. Did you know you have two skeletons in your body? The exoskeleton category includes the bones along the central axis of the body. These would contain bones of the skull, vertebra, ribs, and sternum. The long bones and girdles of the appendages are classified as appendicular skeletal organs. We can also look at bones and classify them based on shape. Long bones are longer than they are wide and contain a central cavity called a medullary cavity. Flat bones are just that, flat. Their spongy bone is often called diploid. Short bones are as long as they are wide and have no central cavity. If these were cells, we would say they are cuboidal. The bones of your wrists and ankles are examples. Irregular bones do not fit into another category. You can consider this classification a junk drawer of sorts. The vertebra would fall under this category. Regardless of the bone, all are made of osseous connective tissue. This tissue contains organic and inorganic components. Now, in biology, organic refers to carbon-containing molecules. The three cell types of osseous tissue are obviously carbon-containing. However, so is the protein ground substance called osteoid. The calcium that is so famous is part of a salt crystal called hydroxyapatite. This crystal contains both calcium and phosphate and makes the matrix hard. Now, depending on the pattern laid down by the cells, we get two osseous tissue types. The outside of every bone contains compact tissue which looks smooth and is very dense. The functional unit of compact bone are osteons, and they are made of concentric lamella with interstitial and circumferential lamella present. However, spongy bones layers form needle-like pieces called trabecula. The trabecula are interconnected and marrow fills the spaces between them. The microscopic structure of this tissue contains these structures. Osteons, the functional and structural unit of compact bone. Lamella, weight-bearing layers, they contain collagen, and they're cylinders. Central canal, blood supply and nerves, these are vertical. Perforating canals, blood and nerve supply that are horizontal. Osteocytes, the mature bone cells that maintain the matrix. Lacuna, small cavities that house osteocytes. Canaliculi, hair-like canals that allow osteocytes to share cytoplasm. While there are 4 categories of bone shapes, we will zero in on long bones. On this diagram, we can see that long bones consist of a shaft, diaphysis, and 2 ends, epiphyses. The epiphysis closest to the torso would be the proximal epiphysis, while the other is distal. Recall that long bones also contain a medullary cavity. With flat bones, we see no cavity and truly a flat appearance. There is a special term for the spongy bone in the flat bone, diploe. Now, all bones have a periosteum. This tissue covers the entire bone except where that bone touches another bone in an articulation. The other surface of the periosteum is dense, regular connective tissue proper, but the inside edge is a thin membrane that contains the osteoblasts and osteoclasts. The periosteum not only houses these cells, but it also carries nerves and blood vessels. The endosteum is an osteogenic layer, the same as the inner periosteum. That travels inside the organ and lies over all the trabecula of spongy bone. Again, we see that it contains osteoblasts and osteoclasts. In the diagram, you can see a 4th cell, osteoprogenitors. They serve as a stem cell that divides and continues to make more osteoblasts. Why? Because once an osteoblast lays down matrix, it becomes an osteocyte. As a result, we actually use up osteoblasts and need a way to replenish that population. As mentioned previously, bone marrow is contained within bones. However, there are 2 types of bone marrow. When we were born, all the marrow was red marrow. Red marrow is hematopoietic, or blood-forming. When we reached puberty, much of the red marrow was converted to yellow marrow. Yellow marrow is adipose and serves as nutrient storage. The remaining red marrow can be seen on the skeleton chart, within the diploe of flat bones, in the vertebra and sacrum, and the proximal epiphyses of the humerus and femur. Use this slide to review the structures listed. Click pause. Review. Now that we have emphasized some anatomy, we'll move on to the physiology of this organ system. There are several processes to review. Osteogenesis creates a new bone. Interestingly, we do not build bone tissue from scratch, but start with some other connective tissue. The process for converting to osseous tissue is called ossification. We'll also look at how bones modify their shapes by remodeling, and lastly, how bones repair from fractures. Long bones form by endochondral ossification, while flat bones form by intramembranous ossification. If you look at the long bones of this colored enhanced x-ray, you can see diaphyses but no epiphyses. This tells us that the epiphyses have not yet formed. Also, looking at the skull, you can see some clear areas between the flat bones. Again, this tells us that these flat bones are not fully formed. The point here is that all bones form over time from a non-osseous matrix. We'll discuss these processes briefly as the details are not part of learning objectives. I feel it is important, however, to know the basics of bone formation. Intramembranous ossification begins with a fibrous membrane formed by mesenchymal cells. Once formed, osteoblasts migrate into the center of the membrane and begin to lay down osteoid and hydroxyapatite as trabecula, thus ossifying the membrane matrix interiorly with spongy bone. Blood vessels grow in between the trabecula. Osteoblasts will then migrate to the periphery and lay down matrix as compact bone. There will remain some membrane on the very outer edges, and that becomes the periosteum. Essentially, intramembranous ossification can be simplified by saying spongy on the inside first, compact on the outside last. Endochondral ossification begins with the cartilage model made of hyaline cartilage. The periosteum first develops on the outside as well as compact bone. Once the compact bone perimeter of the diaphysis is complete, it's called a bone collar, the cartilage cells inside cavitate and die. This leaves behind a cavity within the forming diaphysis. Nutrient foramen or holes are made through the matrix and blood vessels grow into the interior. Once inside, osteoblasts lay matrix down as spongy bone. The forming diaphysis is referred to as the primary ossification center because it forms first. Next, a similar process occurs on the epiphyses, and hence they're called secondary ossification centers. The main difference is that some of the hyaline cartilage is kept. The cartilage on the very edge of the epiphyses will become articular cartilage, and the thin band of cartilage running between diaphysis and each epiphysis will serve as a site of long bone growth during childhood. It's called an epiphyseal plate. Again, we can simplify this by saying bone collar forms, cartilage cavitates. Compact is laid on the outside first, and spongy is laid on the inside last. Now that you have the basics of how the bones form, we can chat about how they grow, change, maintain homeostasis, and heal from damage. Each of these processes will be discussed next. Postnatal growth explains how you grew taller during childhood. Under hormonal control, chondroblasts in that epiphyseal plate undergo mitosis. As they divide, the cartilage here becomes thicker, then begins to undergo the ossification process. Here we can see resting cartilage, then a zone of growth noted by long chains of cartilage cells. Those cells then cavitate, die, and the empty spaces are ossified by osteoblasts and osteoclasts. Note the clear area between the epiphysis and diaphysis of the thumb's first long bone, indicated by the red arrow. Once you can see the structure, you'll see the other epiphyseal plates. This is where growth and length of long bones takes place. Healthcare providers can tell if a patient will continue to grow in height if they see these areas of cartilage inside the bone. Growth spurts occur when we're nearing puberty because the initial rise of estrogen and testosterone trigger more mitosis here. However, once puberty is completed, those same hormones will trigger closure of the plate. In other words, the remaining cartilage here will ossify. At that point, growth in length of the long bones stops. Once ossified, this structure is called an epiphyseal line. Remodeling occurs when a bone changes its thickness or shape. There are two patterns here: resorption, when tissue is removed, and appositional, when tissue is added. Our two cell populations work together in remodeling on the endosteal and periosteal surfaces. That's because that's where they live. The triggers here are hormonal cues, mechanical forces, and even gravity. Note the outline of the original bone in red on the right side. As the bone grew in length, its shape must also change because bones must maintain the proportion of their thickness and size of structures. Clearly, in number 1 and number 3, some osseous tissue was resorbed and removed, while in number 2, some tissue was added. This interplay between add a little here, take a little away over there is remodeling. Bones also remodel in response to mechanical stress or force. Wolff's law holds that a bone remodels because of the forces placed upon it. This is the femur. And you can see that most of our body weight when we stand comes straight down on the proximal epiphysis. Now, if we don't do anything about this, our femur could break right at where those arrows are. To combat this, the femur remodels on the inside. The spongy bone on the interior is built below the stress point. That spongy bone holds the bone up from the inside. In fact, if that spongy bone is dissolved by osteoporosis, the femur may well break at that point the next time a grandma tries to stand up. So how exactly do the osteoblasts lay down matrix? There are many nutritional requirements, but you can see the main items here. Note protein, several vitamins, phosphorus, and an enzyme called alkaline phosphatase. That enzyme helps mineralize bone. Proteins are obviously for collagen production. Calcium and phosphorus are needed for the hydroxyapatite. Now, all of this is not laid down at once. Osteoblasts first lay down the protein ground substance called osteoid in a structure called an osteoid seam. Within 72 hours, the osteoblasts will mineralize the matrix and form a structure called a calcification front. As you know, the resorption of osseous tissue is performed by osteoclasts. The cells are large and form resorption bays on the matrix by secreting acids and enzymes that break down the matrix components. Recall that one of the functions of the skeletal system is to store and release calcium. Calcium is required for more than strong bones. Your nerves need it to communicate with each other, muscles need it to contract, your heart needs it to beat. Strong bones are nice, but they really are a source of calcium to keep the rest of your organs alive. Calcium homeostasis is under endocrine system control. Now remember one of my sayings, the human body always does what makes sense if you let it. So what makes sense now that you know how important calcium is? If you eat extra calcium today, should you just urinate it out? Or rather, should you save that extra calcium for next time? Well, you definitely save it. And by the way, this endocrine system control will be negative feedback. When blood calcium levels are elevated, the thyroid gland secretes calcitonin hormone. This hormone travels to the periosteal and endosteal surfaces through the blood supply. It stimulates osteoblasts, so they respond by pulling calcium from the blood and building new matrix. At the same time, it inhibits osteoclasts so that the new matrix is not dissolved. When blood calcium levels drop, the parathyroid gland secretes parathyroid hormone. This hormone also reaches both cell populations through blood. This time, it inhibits osteoblasts, but stimulates the osteoclasts. The result is resorption of matrix and release of calcium to the blood supply. Here are two more endocrine system homeostasis examples. Growth hormone acts to stimulate mitosis of chondroblasts, resulting in postnatal growth. Growth hormone doesn't always act directly on the cartilage, but stimulates secondary hormone production by the liver. The result is that growth patterns are complex and can be affected by changes in growth hormone or secondary hormone levels. Testosterone and estrogen initially stimulate chondroblast mitosis as well and explain adolescent growth spurts. However, once puberty is complete, these same hormones stimulate ossification of the cartilage, forming an epiphyseal line. In addition, these hormones affect remodeling rates differently. Structured males are generally larger and bones have a greater density. However, structures on bones in females are generally smaller and the bones are slightly less dense. These are not learning objectives, and so I show you these conditions only for interest and application, but not for assessment. Too little growth hormone or secondary hormones produced cause dwarfisms, and depending on the specific issue, there are different types. Here you see two basic categories of little people. However, the opposite happens with excessive growth hormone or secondary hormones. Again, you see two different types of gigantism. So you've learned of the skeletal system histology, how bones form, how they grow, and how they remodel. We'll wrap it up by seeing how they repair from damage. Hematoma formation. When the blood vessels in the matrix are broken from a fracture, blood exits the vessels. But in a simple break like this, the blood is held in place by the periosteum. This allows a large clot to form and inflammation to be triggered. 2. Fibrocartilaginous callus. This is a temporary structure formed by chondroblasts and fibroblasts and will serve as the matrix for osteoblasts and osteoclasts to ossify. This is the functional equivalent of granulation tissue. A bony callus. When the fibrocartilaginous callus is first ossified, the entire structure is laid with spongy bone. You can see the bony callus forming in the X-ray picture below. Remodeling. The last stage where osteoblasts and osteoclasts work in remodeling units to ensure proper structure. Here we see the medullary cavity has returned and compact bone is on the outside. By the way, there's a little extra compact bone here, like a fibrosed scar, that will always be seen on X-ray. This is just Professor Klein being funny.